Hello everyone, my name is Jacob Piland from the University of Notre Dame, and today I'll be talking about our network analysis of synonymous codon usage. First, some background. Many amino acids are encoded for by more than one codon. The different codons that encode an amino acid are synonymous. Synonymous codons are used with different frequencies. Those used frequently are called common codons, and those used infrequently are called rare codons. This is important because rare codons are hypothesized to direct co-translational folding. Furthermore, some rare codons are evolutionarily conserved. We call those that are not conserved non-conserved rare codons, and we call those that are conserved rare codons. In our study, we had two key questions we wanted to examine, and this is the first. Do conserved rare, non-conserved rare, and common codons occupy different positions in the 3D structure of a protein? And for our study, we had the following data. We started with approximately 16,000 protein sequences with rare codon information from a previous study, and we applied the following filtering criteria: available 3D structure, non-redundant sequence, and sufficient amounts of conserved rare and non-conserved rare codon information. This resulted in a dataset of 63 high-confidence proteins. To answer our first key question, we needed to know two things. First, we need to know for each codon whether it is conserved rare, non-conserved rare, or common. Then we need some way to compare the positions of codons. To do this, we convert 3D structure of a protein into a protein structure network, or PSN. In a PSN, a node is a codon, and an edge connects two codons if their corresponding amino acids are close to each other in 3D space. Then we can use graph measures called centralities to compare the positions of codons. We rank codons from most central, in other words, best ranked with a rank of 1, to least central. Now we have labels on which codons are conserved rare, non-conserved rare, and common, and we can compare centralities of the 3D codon types to see if one is more central than another. We designate the relationship of one codon type being more or less central than another with the letter R followed by a number. For example, if conserved rare is more central than common, then that is R1. And there are 6 different relationships in total. Finally, we place a protein into what we designate as a codon usage group based on the relationship it has. For example, if in a protein the conserved rare are more central than the common, and the conserved rare are more central than the non-conserved rare, but there is no difference between the non-conserved rare codons and the common codons, then that protein is placed into group R1, R2. With our proteins thus organized into protein groups that each show a given codon usage trend, we can answer our first question. Do conserved rare, non-conserved rare, and common codons occupy different positions in the 3D structure of a protein? In total, we have 17 different codon usage groups, where all proteins in a group have the same relationships. In this figure, the y-axis is the number of proteins that are in the x-axis, the particular codon usage group. The first group, which we have not covered up until now, is R0, and it signifies no relationship between the codon types of any given protein in this group. 53 of the 63 proteins are placed into a group where there is some relationship. In other words, one of the groups that has at least one or more of R1 through R6, meaning that at least one type of codon is more or less central than another. This would indicate that, yes, conserved, non-conserved rare, and common codons occupy different positions in the 3D structure of a protein. Now we will look at the second key question of the study, which I am introducing. Do proteins with different structures or functions show different codon centrality trends? In other words, is synonymous codon usage linked to protein structure or function? We see that no codon usage groups correlate with sequence. And by correlate, we mean it is not the case that proteins within a codon usage group are more or less sequence similar to each other than they are to proteins in other codon usage groups. In other words, the codon usage groups cannot be formed based on sequence data. However, some codon usage groups do correlate with secondary structure, 3D structure, and/or function. Thus, we provide evidence that different codon types are located in positions in the 3D structure of a protein and may be related to co-translational folding generally. However, further research will be required to more fully understand the role of different codon usage groups in co-translational folding. Finally, I would like to acknowledge and express sincere gratitude for the support of my advisor, Professor Tiana Milankovich, my colleagues in the Complex Networks Lab at the University of Notre Dame, and the collaborators, Professor Patricia Clark, Professor Scott Emmerich, and Professor June Lee. Thank you very much.